So we are now being recorded. Okay, so welcome again to video teleconference number 5 for BIOL 105L YC. In this video teleconference, we are going to look at the calendar on what lies ahead and what's expected in the near future for our subject. Second, we are going to recall the list of exercises we've done so far. And the meat of this video teleconference is a review in a form of a self-quiz for the exercise on photometry of proteins or introduction to proteomics. Right, so this is the calendar. We are now in February and we are currently on time for a video teleconference. Um, However, this is the fifth one, so we're a bit advanced. But I just like to point out that for next week's video teleconference, it actually falls on Chinese New Year. So I guess we have to move our video teleconference next week instead of being on Friday. I'm planning to move it on a Tuesday next week, so a lot earlier. Um, so that's for one. Next is the midterms week is approaching. So on the next video teleconference, I will reveal to you the next group project for the midterm. Okay, so the next group project on the next video teleconference. So these are the schedules so far, and I think I have changed the deadline for the lab report, the current lab report, instead of tomorrow. I think I've moved it on Monday next week. So you have enough time after listening to this discussion. You might want to read again your lab report so you can make necessary changes if there are any. So far, these are the exercises that we've done. And if you look at the list that I've given you, I think we are halfway with our lab exercises already. Okay, so next week there's a new lab exercise on SDS page, still on proteins. Okay, so we are really moving very fast with our lessons. No, so not that it's bad, but I haven't heard any complaint na we are too fast. One exercise per week is too many, so we're going to continue with it. Okay, so all I can do is adjust deadlines. So for this video teleconference, we are going to have a self-quiz on exercise number 5. So I will be flashing questions on the screen, and I'm going to be hearing from you answering those questions, no? Sharing your insights and thoughts. So are you ready for this self-quiz? Do you have your notes ready? Do you have your lab manual ready? Soft copy or hard copy? Do you have a calculator? Because I have a question later on that requires calculation. Ready na? I hope you're not too sleepy after eating that lunch. Are you ready? Let me hear some
Yes, sir.
confirmation. All right. Sige, sige. Okay, so we'll begin. So again, this is not graded. Um, for me, I see this as an opportunity, a fun opportunity to test myself. If I've understood, or not really if I've understood, but if there are certain things that I've missed from the exercise, okay, or if I've understood it correctly, maybe I needed additional explanations. You can also ask uh, amidst this quiz now about clarifications. So question number 1. So which samples did we use to have their protein concentrations analyzed?
Milk.
Yes, okay, that's correct. We had the protein in milk, no? So we had milk samples. If you notice in the video that I've posted, there were two, not necessarily of different brands. And I hope you also took note of the labels in the nutrition facts of the milk because it's written there how much protein they contain. And at the end of this exercise, you are supposed to be determining how much protein per milliliter the milk sample contains so that you can reconcile it with what's in the box label. Okay, so that's the goal of one of the goals of this exercise. So next, which assay was used to analyze the concentration of proteins?
Bradford, sir.
All right, Bradford assay. Okay, so if you've read throughout the manual, it's also said there that Bradford assay is a colorimetric assay. Okay, so meaning to say, um, we base our judgment, on how much protein concentration there is in the sample based on the intensity of a color that's produced from the assay. So it's colorimetric, not calorie, but colorimetric. Okay. So what did we use as a reagent in this assay?
Reagent.
All right. So it's also known as the Bradford reagent, but what is it really? There's another name for the Bradford reagent. It's actually a dye. So what's the name of the dye?
Kumasi Brilliant Blue, sir.
Yes, that's correct. So it's Kumase Brilliant Blue. Okay, so which we also call as the Bradford reagent. So the name Brilliant Blue actually is misleading because by itself alone, it's not blue in color. Now, if you notice in the exercise, it's not really blue when I placed it in the cuvettes. However, the resulting color when there is protein will be brilliant blue or dark blue. That's why it's called Kumase Brilliant Blue. No, that's when it embodies its name when there is protein present. I think we've answered number four. When there is protein, the sample would look blue. The darker the blue is, the more proteins there are supposedly in the sample. All right. How about when proteins were absent? What color will the dye be?
Gray, sir.
Gray. Perhaps it's close to gray, but what is the description? When it's gray, there's actually a bit of protein still. But with the complete absence, what's the color of the dye?
Light brown.
All right. So reddish brown or brownish, okay, would be the color of our sample with a dye in it if there was no protein. So really the Kumase Brilliant Blue dye is not blue by itself. But when there's protein, it appears blue. So by itself, it's reddish brown or brown. Okay, so before we proceed with the next question, what is the reaction? Can you describe how the Kumase Brilliant Blue reagent, the Bradford reagent, how does it react with the proteins? On which part of the proteins will it get attracted to?
Based sa manual, sir, kay with the side chains or are groups of specific amino acids.
That is correct. Okay, so specifically, it reacts to proteins because of the presence of the certain R groups in the amino acids that are found in the polypeptide or the protein. So that's correct. So when that happens, the protein and dye complex, we now call it protein and dye because they stick together. It's a complex. It will appear blue to our eyes. So thus, the change in color. So what did we use to measure the color change or the intensity of color? Because if you can remember, you were asked to describe the colors, compare the colors using your eyes. Okay, that's part of the step. But then we needed an instrument to really measure the actual color. What was the instrument?
Spectrophotometer, sir.
All right, so we used the spectrophotometer. And what value was measured when we used the spectrophotometer?
595 nanometers, sir.
Uh, Technically, you're correct. What did we measure, rather? That's correct. What did we measure?
Absorbance,
Absorb.
sir.
You are both correct. Okay. The absorbance of our samples, no? And you already mentioned the number. Okay. So we'll see what's next for the questions. Okay. So this one, which color and wavelength did we measure the absorbance of the sample?
595 nanometers, sir. Yellow light.
All right. So that's a yellow colored light, no? Yellow light. So our spectrophotometer is actually able to emit specific colors and wavelengths of light. You just need to set it. If you can remember in the video when we set the spectrophotometer, I typed there 595 so that it will specifically emit the light with 595 nanometers of wavelength, the yellow light. But I have this question, why yellow light? Why 595 nanometers? You said we were measuring absorbance. Then specifically the absorbance of yellow light. So my question is, why yellow light? Okay, let's go back to the color of our sample. When protein is present, what will be the color of the dye and protein complex? What will be the color again?
Blue.
Blue. Okay. What will happen if we use blue light? Will blue light get absorbed by our blue sample? Yes or no? When you look at the colors of leaves, many leaves are green in color. Does that mean they absorb the color green light?
No, sir.
No. So our blue samples with the proteins, if we use blue light, does that also mean that they do not absorb blue light? I think you know about this, diba? The color of the object. When I'm looking at something red, what light is it reflecting? What color?
Red.
Red. So if our blue sample appears blue to our eyes, that means it's reflecting blue light. So if we use blue light in our spectrophotometer and our sample is blue in color, we will get zero absorbance. So we're doing the opposite. Okay, because blue na nga ang color ng specimen mo, tapos you're using blue light, it will just be reflected or it will just pass through the blue color of the specimen. So why yellow light? Back to my question in number 9. What is the peak absorbance of a blue colored object? What color will it absorb? It's actually in your manual. The peak absorbance of the blue colored dye and protein complex is the answer in number 8, which is 595 nanometers or yellow color of light. So that's the reason why we will use yellow colored light, because we know if our sample has more protein, the bluer it is, the more yellow colored light it absorbs. And the less blue it is, the less yellow colored light it absorbs. Now, so we use the yellow color. Okay, so that's for number 9. I hope you were able to think through that. Do you have any concerns about that one? Any questions?
No, sir.
Okay. So for example, if you see a plant, it appears green, it reflects the green color, and it absorbs mostly all the other colors aside from green. Okay. Mostly red, the red color, no, is used in photosynthesis. So you will learn that in our next lesson in the lecture about photosynthesis. So here, number 10. What was the purpose of the protein standards? If you can remember, there were two samples, A and B. Then there was a blank. Then there were 7 protein standards, 1 to 7. What's the purpose?
I believe, sir, the purpose of protein standards are their properties have already been predetermined, so they're used as a reference, sir, in order to help uh, compare and identify the proteins in the sample being studied.
That is correct. No? And when you mentioned that their properties have already been known, that's exactly correct. Their colors are known. And most importantly, their absorbance of the yellow colored light are also known. Okay? Or will serve as a standard later on when we measure it in the spectrophotometer. So that if you can remember, one of the activities in the exercises, you need to make a standard curve using the absorbance of these standard protein standards. Okay, so without the protein standards, you won't be able to determine the actual concentration of proteins in our milk samples. What about the blank? No, the blank, the cuvette labeled blank. What was placed in that cuvette?
Bradford
PBS,
reagent only, sir. I did
PBS,
All right. Continue. Bradford reagent, kulang yun.
and PBS.
Okay, correct. So you have the PBS, you have the Bradford reagent. What's missing will be the protein content. Okay, so in other words, this is a negative control. No, so it's something, a setup that doesn't have what we are measuring, so that we would know the baseline if the protein is absent. So that's the one labeled blank. Okay, that's correct. Next, um, I think I already mentioned the answer to this. The reason why we have a blank because it's a negative control so that we'll see the color and we'll know the absorbance when protein is absent, no, from the sample. Not only that, there's another reason. What's another reason for the blank? Sige, anyone? Aside from a benchmark for ourselves, why do we need to put a blank? If you can remember in the spectrophotometry method, I placed the blank first, right? In the container in the spectrophotometer. And then I pressed 595 and then I measure the blank, its absorbance. What was that um, procedure called, or what is that procedure for?
Calibration, sir.
All right. So calibration. So it's very important in that particular method to have something as a baseline. And your spectrophotometer will not know that one. You yourself will be the one to give it instructions. And we do that by measuring the absorbance of our blank first and then setting that as the zero. And then that's the baseline for everything else. Okay, so blank will be for calibration as well. So someone answered this already a while ago, but what is it that we use to dilute the samples, the milk?
phosphate buffer saline, sir. PBS.
Yes, okay. So we cannot use the milk as it is milk because it's white in color. It's going to affect our results, no, when we measure absorbance because it's white. So we need to at least dilute it with something that's neither acidic nor basic. So that's PBS. And what is the dilution factor? How much milk was added? and how much PBS was added?
1
40.
is to 50, sir.
All right, 1 is to 50. Someone was saying something also. Is it the same answer?
Yes, sir.
1 is to 50, and that means um, for every 50 microliters total volume, 1 microliter of that will be the sample. So in our case, it was 200 microliters, if I'm not mistaken, of the whole volume. So you need 196 microliters of the PBS and 4 microliters of the milk sample. So that's 1 is to 50. Or because the question is dilution factor, that's 50. 50 divided by 1. So 50. Okay, so prepare your calculators. A sample was found out to have a protein concentration approximately equal to protein standard 2. So what is its protein concentration in milligrams per milliliter? Okay, I'll give you time to calculate.
Is it 0.250, sir?
That's the concentration of protein standard 2. But the question now is milligrams per ml. What will be the concentration now? Considering that we use the same method as we did in the exercise. Sorry. So correct yon for protein standard 2. But if we did what we did in the exercise for the milk, what will be the answer in number 15?
7.7 milligrams per ml, sir.
How did you solve for that?
Get the absorbance value of the protein standards 2 and then multiply by the dilution factor.
Okay. Almost correct. You do not multiply the absorbance to the dilution factor. Baliktad. You multiply the protein concentration of the standard to the dilution factor. So what will be the answer?
12.5 milligrams per ml, sir.
That is correct. Okay? So sa exercise kasi, yun yung in the exercise you get the absorbance, right? Okay. So once you get the absorbance, you compare it to the absorbance of the standards and to the closest to the one to where it's closest to among the seven standards, that will be the concentration in milligrams per ml of your sample. And then you multiply that by 50. So that's how you solved it, hopefully in the exercise. Okay, you don't multiply the absorbance to 50. You multiply the concentration to 50. All right. That's for the review. So now I would like to just clarify how the spectrophotometer works. No? So a while ago we said that because our protein and dye complex appears blue, then it must be absorbing more of the yellow colored light. No? So as we can see in this visible spectrum here of light, the value that we set our spectrophotometer to emit was 595 nanometers, and that's about here, and that's yellow colored light. Okay, and the main reason is because blue colors absorb yellow colors. Okay, so once you type that in your spectrophotometer, oh, I want it to emit 595 nanometers, the spectrophotometer is capable of choosing which light to emit. So in this case, in this illustration shown here, okay, this spectrophotometer has been set to emit only 595 nanometers, yellow colored light. So without any sample, you see that the light hits the detector, and the detector will be able to detect, oh, this is a lot of yellow light. Okay, so what will happen if we put a sample? Okay, let's say a blank. Okay, so now that we've placed a blank in between the incident light, no, and the detector, since the blank is not blue, then it will not absorb most of the light. Meaning to say, most of the light will pass through, and the detector will be able to detect, oh, there is still some light passing through. The more light which is transmitted there is, the lesser absorbance value there will be for your sample. Okay, so that's for your blank. So now you can tell the spectrophotometer, oh, make this zero absorbance. This will be my calibration now. Okay, this is zero absorbance. Now, after you've calibrated your spectrophotometer, you might want to put in a sample. So this is number 7, standard number 7. And because it's very blue, it's going to be absorbing all of the yellow light emitted by the spectrophotometer, and no light is going to reach the detector. No transmitted light. And because the detector doesn't detect any yellow light, the spectrophotometer will tell you that the absorbance is very high, 1 point something. Okay, 1 point something absorbance because no light was able to pass through the sample. Okay, so this is just a simple illustration. No? So we're talking about absorbance, but there are other quantities that the spectrophotometer can actually measure. It can also measure how much light can pass through. Okay, so if absorbance is A, there is another value that the spectrophotometer can measure, and it's called T. What do you think that is? If A is absorbance, you can use a spectrophotometer to measure that. It can also measure T. What is T? It's already written here on this slide. What is it?
Transmitted light.
Yes, okay, transmitted light. So if we were to measure transmittance, we call it transmittance, opposite ang magiging resulta. No, your control will have a lot of transmitted light, and your standard number 7 will have zero transmitted light, opposite. But since because we measured absorbance, our control had zero absorbance, and our number 7 standard has 1 point something absorbance. Okay, so this is how a spectrophotometer works. Now, and then there are other uses. You can even measure the absorption spectrum of certain pigments like chlorophyll using the spectrophotometer. So in the next lesson in the lecture, you will be able to see what an absorption spectrum is. It's part of the lesson also. Okay, that's about it. Are there any questions about the spectrophotometer and how it works or about the exercise? Questions about the concepts? So I believe you were able to understand the concepts of absorbance and how it may be used as a colorimetric assay to tell how much concentration of protein there is, no, in a particular sample. No, we call it the Bradford assay. Okay. So with that, because maybe you haven't thought of a question yet, here's my announcement. Exercise 5 is due on February 8th. That's Monday next week instead of tomorrow. So you have more time. If ever you might need to edit some things, you can still do so. Okay, and that ends my presentation. So it's now your turn to ask questions. No questions? Okay, that's good. Okay, that means you understood. No concerns. Okay, I like that one. So keep it up. Okay, but if ever you have questions later on, merong magko-question. If you have questions later on, you can always message me in eLearn or in Messenger. Okay, and I will answer you as soon as possible. So last
Thank
chance. Yes, last chance for questions in this video teleconference.
None, sir.
None. Okay. So thank you very much for listening and for sharing your thoughts. Um, what do you call this? Thank you for attending this video teleconference. And a recording of this video teleconference will be posted later on for you to review if you need to do so, and for your classmates as well who are not here. So have a productive day ahead, have a safe day ahead, and you are now all dismissed from our meeting.
you, sir.
Thank you,
Thank
sir.
you, sir.
Thank you, sir.
Thank you, sir.